This is our second part of looking at the regulation of the cell cycle. So let's get right into it. Well, actually, let's just kind of think a bit more generally to start with before we get carried away. Let's see. The cell cycle is largely controlled by the phosphorylation of target proteins. That would be done by protein kinases, and also the dephosphorylation of certain proteins by protein phosphatases. So phosphorylation and dephosphorylation are two of the main mechanisms. There's also a third, protein degradation, and we'll take a look at that a little bit later. So there's a special family of protein kinases that are they're only active when they're bound by another molecule, another protein molecule called a cyclin. And so these kinases are called cyclin-dependent kinases, or the CDKs. So cyclins are called cyclins, and they were actually named by my PhD supervisor, who discovered them many, many years ago and won the Nobel Prize in 2001 for their discovery. They're named because their abundance oscillates during the cell cycle. And so as you look at different types of cycling molecule, there are several. You can see in the bottom graph here, there's A, B, D1, E, D2, and then a number of very specialized cyclins. You can see that their levels vary as a function of the cell cycle. So we have mitotic cyclins, which are cyclins A and B, and they're required for the G2 to M transition. They bind to mitotic CDKs, and that complex of mitotic cyclins and mitotic CDKs is called MPF. Now that's often, and I suspect in your book, it's often called mitosis promoting factor, and it's actually wrong. It's not. It's called maturation promoting factor because when my boss discovered these cyclins and subsequently this activity called MPF was discovered, they were working in frog eggs, and they were looking at the role of this protein kinase activity in frog egg maturation. So it's not mitosis promoting factor, although that's sometimes called mitosis promoting factor. Actually stands for maturation promoting factor. Anyway, so that's the mitotic cyclins. Then there are G1 cyclins that are required for passage through G1. So things like cyclin D1 and cyclin E primarily. And they get, cyclin E mainly gets the cell cycle past an important point in G1 called the restriction point. And that's the point where cells decide whether they're going to commit to a round of cell division or not. So cyclin E is very important in the cell's commitment at restriction point to a round of cell division. Those are called G1 CDKs. Then you have the S-phase CDKs, and those include partly cyclin E, partly cyclin D, and partly cyclin A. So there are several S-phase cyclins, and they're required for DNA replication during S-phase. So we've got one level of regulation of the CDK by the cyclin, and a second level of regulation of the CDK occurs via phosphorylation of the cyclin-dependent kinase. So we have, it's interesting about mitotic CDK. So mitotic CDK, the kinase, has got a constant level. So if we put the actual cyclin-dependent kinase on here, you have a constant level up here somewhere throughout the cell cycle. But it's only active when it binds to a cyclin A or a cyclin B. And those concentrations increase as we move through the cell cycle, through S phase and G2, until we get to a peak of those activities at the end of G2 and M, and that triggers the entry into M phase. So the mitotic cyclins reaches critical threshold at the end of G2, and that permits the CDKs to trigger the onset of mitosis. And then what you see is that there's a rapid destruction or degradation of cyclin A, and then in the middle of M phase there's a rapid degradation of cyclin B. So that's critically important in getting cells, particularly B-type cyclin degradation, getting these cells out of M phase. And so we'll look at the exact point and mechanism of degradation of a B-type cyclin. So a lot of people study the B-type cyclins. I spent a number of years looking at cyclin B and cyclin A. And so B-type cyclin, the first one discovered in the role of mitosis, is called the mitotic cyclin. Okay, so let's take a look at the phosphorylation-based regulation of CDK activity. So there are both phosphorylations involved in the activation of CDKs as well as dephosphorylation. That sounds a little bit kind of oxymoronic. So Let's take a look at it. We've got a mitotic CDK. We're thinking about like B-type cyclins here in mitosis. So we've got a mitotic CDK, which will just be abundant throughout the whole cell cycle. And then as the mitotic cyclin, the B-type cyclin starts to accumulate, it is able to associate with the CDK. And so that's called the mitotic CDK cyclin complex. And that's inactive. Doesn't do anything. There's no kinase activity at all. So you can isolate this from cells, and it's got no activity whatsoever. Then there are two inhibiting kinases. And so an inhibiting kinase is a kinase which adds a phosphate to a target protein, and that interferes with the architecture of the active site, and so effectively kind of blocks access of substrates to the active site of the CDK. So these inhibiting kinases put two phosphates onto the CDK, and then another kinase, an activating kinase, adds on another phosphate, and that's going to turn the kinase on. But it only turns the kinase on when you dephosphorylate these two original inhibiting phosphorylations. So you've got two phosphates that go on that turn everything off, and then you've got one that goes on that turns everything on only when you take these first two away. So the first phosphate goes on, you add you put the first two phosphates on, that turns everything, keeps everything off. You add the third one, nothing happens, and then you take these two off. Oops, sorry. You turn those two off, and then everything becomes active. So that phosphatase there is removing these two phosphates that went on to start with over here, and that gives you a highly active mitotic CDK cyclin complex, and that's what will drive the cells into mitosis. And once this is formed, it actually stimulates this phosphatase here. So this is one of the rare places in biology that you actually see a positive feedback loop. So positive feedback loops are where the outcome stimulates the events which lead to the outcome. So blood clotting is a good example. When you have blood clotting, you don't want it to stop halfway through. You want it to keep going. Childbirth is another example. And so once you've started initiating the passage into mitosis, you don't want to suddenly stop and back out again. You're committed at that point. And so there's a positive feedback loop which drives the cell into mitosis. So that's kind of an interesting little phenomenon. So what do these kinases do? So let's just keep thinking about the mitotic CDK cyclins to start with. So once they're activated, they do things like they phosphorylate a bunch of target proteins, and the subject of my PhD thesis was trying to find a lot of the substrates that these mitotic cyclins and CDKs phosphorylate. And I also worked on cyclin A, which works down here in S phase. So I was interested in the kinds of things which are phosphorylated by CDK2 and by the S-phase cyclin A. So, for example, mitotic cyclin CDK complexes phosphorylate the lamin proteins of the nuclear lamina, and that causes the nuclear lamina to break down, destabilizes the nuclear envelope, so you get fragmentation of the nuclear envelope, so you can do so you can form the spindle and have the chromosomes released so that you can bind the microtubules to the kinetochore. They also phosphorylate a protein called condensin, which is involved in chromosome condensation. And so that helps to get the chromosomes condensed so they can be segregated. And then there are a number of microtubule-associated proteins which are phosphorylated to allow the assembly of the mitotic spindle. You also, once you go into mitosis, the cyclin CDK complexes also activate a complex of proteins called the anaphase promoting complex. That does exactly what it sounds like. It basically allows the cell to go from metaphase, where the chromosomes are lined up on the plate, into anaphase. And so this anaphase promoting complex is what that does, is it adds ubiquitin, which is a small 9 kilodalton protein. It adds ubiquitin onto mitotic cyclins. And so when APC, the anaphase promoting complex, adds ubiquitin to the mitotic cyclin becomes degraded, and that's the trigger to get cells through anaphase. So if you can't degrade the cyclin molecule, you get stuck at metaphase, and cells just sit there. So to get through anaphase and the rest of the mitotic process, you've got to take away the mitotic cyclin. Now the good thing about this is it makes this unidirectional. That means there's no going back. Once the cyclin is gone, it's not going to accumulate again until you're well into the next cell cycle. So once you've triggered entry into into mitosis, you've got that positive feedback loop, which drives you in and stops you going backwards. And then you degrade the cyclin, and that stops you going backwards as well. So two main steps there that make sure this is a unidirectional process. You only go forward in the cell cycle, there's no going back. And so we'll look at some processes in a little bit which allow the cells to check whether everything has been done before they go forward into the next step. And those are called cell cycle checkpoints. So we'll take a look at those. So that's a little bit about the activity of the mitotic cyclin CDK complexes. Another target, one of the probably more interesting ones, from my opinion, interesting is subjective. One of the more interesting proteins from my perspective is a protein called Securin. And Securin is a protein which is intimately involved in sister chromatase separation. So Securin is here, and it is an inhibitory protein of this enzyme called separase. So what separase normally does is it cleaves a kind of molecular glue called cohesin. And cohesin keeps the sister chromatids stuck together during metaphase, up to the point of metaphase. So when the chromosomes are replicated, they get stuck together. Sisters get stuck together by this protein called cohesin. And this protein called separase basically degrades or chops up the glue to allow anaphase, allow this business to happen. And so that's protein Securin grabs hold of separase, and separase, when it's bound to Securin, is inactive. And so when you activate APC with the mitotic cyclin and CDK complex, you phosphorylate APC. APC degrades Securin. So if we take Securin away, we're taking away the inhibitor of separase. So that means that separase can work on cohesin. And so the released separase degrades cohesin, and that means we can do the metaphase to anaphase transition. So what we've got is we've got the mitotic CDK cyclin complex targeting a number of substrates which regulate entry into and passage through the cell cycle. So they're also involved in exit from mitosis and cytokinesis, but the primary activities I want you to focus on are these activities of getting us into and through the first parts of mitosis. So when you take away the cyclin, you take away the CDK activity, and it's the loss of cyclin and CDK activity which allows you to do things like reassemble the nuclear envelope. Remember that the CDK cyclin was phosphorylating like the nuclear lamins, and so if you take away the phosphorylations on the nuclear lamins, the nuclear envelope reforms. If you take away the enzyme activity, which is responsible for the condensation of the chromosomes decondense. So lots of things that are required on the degradation of the cyclin as you go through to the end of mitosis. So we'll wrap it up there. And we'll come back and we'll start looking at some of those checkpoints. I'll see you soon. Bye for now.